or the other area, which is wastewater treatment. And a chunk of what I'm going to talk about has just recently been accepted for publication. And then there's a whole lot that I've never presented before. A lot of it is my own data analysis. And I'm looking for some feedback on that. I'm taking kind of a risk of putting it out there because I don't know if I'm doing it right, but I'm hoping that this is the right community to get some feedback on that. So I do study bacteria in water. That's kind of the common theme. On the top there, I show 2 bioreactors that we culture bacteria that are really good at storing phosphorus inside their cells. And so the engineers that are in my lab, a lot of them want to work on something that, you know, involves pumps and timers and, you know, chemostat vessels and get some of that practical engineering experience. But we still do a lot of genomics, sort of kind of systems biology, you know, metagenomics, metatranscriptomics, with those bacteria that we isolate. We can't grow them in culture, so it's a challenge to do that work with just an enrichment culture, but we have figured out some strategies to get it really enriched. And I hope that someday I get to tell you about that because it's a really neat system. But I'm going to be talking today about the freshwater lakes that we study. And we like lakes as a model system for studying microbial communities because they are sort of these defined islands, really. If they have rivers coming in and out, then of course there's communication with other bodies of water sometimes. But they're really important for things that people who value water quality, you know, for recreation or for drinking. Microbes are really important for determining that water quality and the ecosystem services that lakes provide. And a lot of the ecosystem scientists that I collaborate with don't really care about the genes and the bacteria. In fact, their eyes just glaze over when I start talking about genomes. They want to understand how the microbes are interacting with the chemicals in the water and the higher level food web organisms like fish. And so I'm kind of always there advocating for why it's interesting to study them, you know, at the level of their genomes, which is kind of my role in that collaboration that I have with the folks over there in the Center for Limnology. And so I've had a long relationship with the North Temperate Lakes Ecological Research Site, which is in the Center for Limnology, and it's a really amazing resource here on campus. It is an NSF-funded program that goes back to the 1980s. It gets renewed every 7 years, I think. And it supports a lot of other research projects by giving us access to field equipment and chemical analyses that we couldn't do ourselves, and just sort of a community of researchers who study lakes. And one of the things that I learned from the researchers over there very early on was this concept called the invisible present. And it was developed by the first PI for the NTL LTER. His name is John Magnuson. He's still around, retired, but still around. And he framed it using ice cover on Lake Mendota as the example. So this is from an old paper, as you might be able to tell from the quality of the image, but it shows you the duration of ice cover in 1 year, so 1983. So this is ice cover on Lake Mendota. And if you look at it at that 1 year, you might say, well, you know, it's about 50 days that we have ice cover here in Madison on Lake Mendota. But if you expand that window a little bit more, you see that that's actually kind of an anomaly. It's a low point in the distribution of how many days ice is on the lake. And as you expand that window farther and farther, you see, you know, that there's low points and high points. And here you can't really see a downward trend yet, but you start expanding it out farther, and you can see that not only do you have the interannual variability kind of sub-decade level, but then you start to see the downward trend that's continuing as a result of climate change. And so this concept of the invisible present, that the window over which you're examining a natural ecosystem and the length of time that you have been observing it, really matters in terms of the patterns that you perceive, and then of course the processes that create those patterns that you try to understand. And when you're trying to study something like a lake that's experiencing all these different drivers like a long-term climate change signal and land use change signals. We have 2 invasive species that have come into the lake since I've been here. So we want to try to understand the complexity of all of those drivers, and we need to be able to watch it over a long time. And so I was extremely fortunate that when I walked in the door here into almost exactly 20 years ago, there was a professor in the agronomy department who was the PI of what he called the North Temperate Lakes Microbial Observatory. So back then, the NSF funded 5-year projects to intensively study ecosystems from the perspective of microbes. And they only did, I think it was 3 rounds of funding maximum, and then they shut down the program because they figured we'd observe microbes as much as we need to. But he had this grant. He decided he was leaving UW-Madison to be a department chair somewhere. And he called me and he said, Trina, first thing, welcome to campus. Second, beware of full professors bearing gifts. Third, do you want my grant? And so he gave me this grant that was like, it had like 3 years left on it and a postdoc and 2 grad students and an archive of samples, only about 3 years worth of samples. But we kept it going, and it was an extremely, serendipitous thing big advantage for me to get that right at the beginning. And we carried it through. I mean, we still collect samples now. The data set that I'm going to talk about was analyzed up until 2018. There's a few 2019 samples in there. This was the deep love of a PhD student named Robin Rower, who graduated in 2019. She pulled all these samples out of the freezer and extracted the DNA from them. These are water samples that we collect on a filter, and then sent them all out for 16S ribosomal RNA gene sequencing. So I'm going to tell you a little bit about what she found with that. And that's the work that's published now. And then she also sent them out for full metagenomic sequencing. So almost 20 years, hundreds of samples with varying amounts of resolution through time, but sort of really an unprecedented scale of, 2 decades plus the resolution that you need in order to see changes that are happening in the microbial communities. And so, the first thing that a microbial ecologist does when they look at a data set full of 16S ribosomal RNA genes is to make an ordination. So if you haven't seen an ordination before, it's just a multivariate statistics technique to visualize how much one data point is different from another data point. So if 2 points are close to each other, then they're more similar than points that are far away from each other. So each point is a sample, and the distance between the points is based on the community composition, how different one sample is from another in terms of its 16S ribosomal RNA community composition. And if you haven't heard of that before, just think of it as a way to count species, right? We can't count microbes by using plates because they don't grow, most of them. And we can't see them and count their legs and arms and whether they have hair. So we use a barcode which is the 16S ribosomal RNA gene. And so it was too much data to put on one ordination. So she put it onto 3 and has the fall and spring communities here on the left. And you can see they were broken out here by, these distinct time periods in the year time span of a lake. And you may not know what clear water is. That's just a term for an early spring condition where the water is very clear, and it's because of zooplankton who graze on all the algae. But the thing that you should notice is that the points are clustering by color, which means they're clustering by season. And all the years are on here. So like all the years are overlaying on each other for any particular season. So that tells us that it's reproducible across years. And we call that a phenology. Maybe you've heard of Aldo Leopold walking around with his little observation notebook saying that, the birds all come at this time of year back from their migration. So it's the microbes all come back, ready for you to go sit on the terrace and watch them in the summer. And we see that recurring pattern, which wasn't all that surprising. I mean, we've seen that before in shorter time periods. So that was kind of just a gut check of like, are we seeing the same thing when we have 19 years of data that we saw when we had 6 years of data, sort of that invisible present concept. And so I think one of the amazing things about this data set is that, we see those repeated patterns, but there was actually this really amazing natural experiment happening at the same time that we knew about, but we didn't really have any concept that it would affect the microbial communities. This is a lot of work done by my colleague, Jake van der Zanden in the integrative biology department, who's also in the Center for Limnology. And he and some undergrads actually discovered that there was an explosion of spiny water flea in the lake in 2009. So spiny water flea are predatory zooplankton that eat the native zooplankton and really mess up the food web and nutrient cycling in the lake. And so they, got a foothold and seemed to persist through the next 5 years or so. And then we had a zebra mussel invasion. And hopefully Carol can figure out what's going on there. But basically, we know that they were in the lake a lot earlier than that, but they didn't really erupt or like get, a big enough population size to really see them until about 2015. And so we have, in our data set, we have pre-2009 as kind of the baseline conditions before invasion. And then we have, during the spiny water flea, but pre-zebra mussel and then post-zebra mussel. And what Robin found when she analyzed those data was that there does seem to be an effect of those invasive species on the cyanobacteria that are in the lake, which are often the metric that people care about, right? Like how blue-green is my water? How toxic is it going to be if I go swimming? And so what we found is that the cyanobacterial communities are appearing in the lake earlier, so their blooms are starting earlier, and they are persisting longer into the fall. And that holds both post-spiny water flea invasion and post-zebra mussel invasion. And she also found that the diversity of the cyanobacteria is increased post-invasion, post-spiny water flea invasion. We don't really have enough data points or years after the zebra mussel invasion to really separate out, whether or not that diversity pattern is holding or not. But just to make sure that everybody's on board with what the axes are here, this is a time zero that we define based on when the summer starts, which actually shifts around. It's not an exact date. It's based on water temperature and other things that change a little bit year to year. And then the y-axis is just species level diversity, if you want to think about it that way. So it's an index that accounts for both richness and evenness. So what you should notice is just that the post-spiny water flea cyanobacterial communities are more diverse in kind of the high summer period. And we don't really know what the consequences of that are in terms of just like, what does it mean that there's more different species of cyanobacteria? But we have also some data about the toxicity of those cyanobacterial blooms. The cyanobacteria make toxins, which is one of the reasons why we worry about them being in the lake. And the toxin compositions and abundances have also shifted as a result of that shift in the cyanobacterial communities. And so this is kind of the community ecology and water quality type of questions that we can ask with this long data set that have real implications for the stuff that people care about in the lake. That kind of a community ecology and counting species and how diverse they are and how they change is what we've done in the lab for 20 years. It's kind of like what I'm used to doing. But what's amazing about this data set is that we can go a lot deeper into what each of those species are, like what kinds of traits they have based on their genomes, and what level of diversity do they have at different scales, which will become, what I mean by that is a little tricky, but we're going to walk through it. So the things that we're counting in that work I just showed you that showed this very clear shift in the cyanobacterial communities is all based on 16S ribosomal RNA. And the acronym that we use now is ASV. amplicon sequence variant, I think is what it stands for. But, you know, we like to pretend that it's at the species level. That's the way we count. You know, we do Shannon diversity. We look at beta diversity based on, you know, standard community ecology metrics that need species that you're counting. But there's a lot of debate in the field of microbiology, which some of you may be very aware of what is a species and how do we define it? And people get very uptight about that. We have like entire conferences dedicated to thinking about this. And now that we have genome sequences that we can easily get, I think people are really starting to question a lot of that 16S based data. I think there's still it's still a powerful technique and you can answer a lot of good questions with it. And we've learned a lot with it, but now that the genome sequencing is as cheap or sometimes at scale, even cheaper, that we can start to look at genomes. And so if you think of a genome as a collection, think of a species as a collection of, you know, diverse entities, you know, microbiologists might break them up as, you know, subspecies. And then we use the word strain. And then you can argue about what a strain is. Is it an isolate? Well, what if it's not isolated? Can you talk about strains in a natural community where the organism hasn't been isolated before? Even within a strain, if you grow E. coli in a flask, like you get mutations and you get diversity. You know, the Richard Lenski experiment that has looked at E. coli over many thousands of generations shows that really beautifully. So there are going to be multiple genomes that represent a strain. And if you want to, you can get down to the level of a single genome, which maybe that's a single cell, right? You know, now that we have single cell genomic techniques, you can access, you know, that, you know, exact genome sequence. It's not a fabulous technology in the sense that it's hard to do. It's expensive. Only a few people do it well. And you don't ever get a complete genome anyway. So you're always missing something just by the nature of the technology. So we can scale across those different levels of resolution and ask, well, where do these ecological patterns show up? Like if we start lumping versus splitting the genomes, does that affect sort of the patterns through time or inferred interactions between species or between strains or however you want to define it? And so once you have the genome sequences, a lot of new questions, you know, jump into your head about what you want to analyze. So I'm going to come back to what a MAG is in a second. But we went back to that same sample set that Robin did the 16S sequencing on. And we sequenced, you know, not quite all of them, but still a lot. And we did this in collaboration with the Joint Genome Institute. We got one of those community sequencing program awards to do up to 10 terabytes of sequences. So we worked with them a little bit, but did a lot of it are doing a lot of it on our own. If you're not familiar with the concept of genome resolved metagenomics, well, first, who has heard of genome resolved metagenomics? Woo, okay, good, then we'll go through it. So this is actually kind of an old figure now, it's from 2015, but the authors were kind of pioneers in this method. Basically, you get a sample from the environment, you extract the DNA just like you would for 16S sequencing, but you don't do PCR. You just shove it through a Illumina machine. You know, back in the day, we used 454, but that wasn't great. So now we use Illumina. Some people are now shifting to Oxford Nanopore and getting, you know, long reads. Most of our work is done with Illumina now. So like paired end reads about 200 base pairs, maybe 150 base pairs, depending on the run that we do. So that generates a bunch of reads, right? So sequences that come out of the sequencer. And you can then, you know, shove that through a bunch of informatics workflows, some of which are done at the Joint Genome Institute, some of which we run ourselves. And you assemble all of those reads into contigs, which are now longer, right? Because they're the stitched together reads, but they aren't sorted by which organism they came from. And so on this figure, there are different colors based on what genome they came from, but obviously they're not in the text file that you get after you run the assembler. So then you have to sort them like you would Legos. And we call that process binning. And the real breakthrough in genome-resolved metagenomics happened probably in around 2017, where someone, this group in Denmark figured out that you could use differential coverage of the contigs combined with genomic signatures that, most often it's tetranucleotide frequencies. So you compare the signatures of the different oligomers or k-mers in the contigs and how much coverage they have in your sample. And you use those two to sort them and say, I think all of these red contigs came from one genome. And so you put them in a bin, right? And you do that with all of your contigs, as well as you can. And then you have what you consider to be a draft genome. And sometimes it's, a really high quality genome. It's like estimated to be 90% complete and like, 0.5% contaminated with something else. And you're like, that's awesome. More often you get about like 50% complete. And we tend to get lower completeness, but not very much contamination. So the way that the workflows are going, it seems like it's good about not accidentally putting in a contig that doesn't belong in there, but you miss some. So it's kind of conservative that way. So the informatics workflows are getting much more mature and accessible. So, you still need a big computer. You need big computers. So that's been a bit of a struggle getting the computing resources that we need. So it's not, something that anybody can pick up and do. You need to go and get trained on how to use those workflows. But it's getting better, and it's getting to the point now where, if you go out and do a bunch of metagenomes, you're not going to publish that paper in Nature anymore. You might get it into ISME Journal or, MBio or something, but like it's you got to do a little bit more these days than just generate a bunch of genomes. And what we get out of this is usually a collection of metagenome assembled genomes, which is where MAGS comes from. And it's just a collection of genomes. Imagine that you downloaded a bunch of isolate genomes from the NCBI GenBank, and now you can use that for doing comparative genomics. And you can define what quality cutoffs you want to use. Like, I'm only going to use genomes that are at least 75% complete, and those kinds of things. And so, there's still a lot of debate in the field about how to do that, but we're going to move forward here, hoping that you trust me that I made some decisions that are okay. I'm not going to talk quite yet about the yellow part down there. We're going to come back to it, but here's what okay. So this is where we transition into stuff that I have done myself, which, professor doing actual research is a little scary, but it's sort of my hobby. And this is a collection of genomes, and I don't expect you to read the names. But what I want you to recognize is the shape of the rank abundance curve, right? So there are some genomes that are really abundant in the data set. So this is just a measure of what is the coverage of that genome across all of those 460 something samples, right? And you can see that there's some that are very highly abundant, and the most abundant one is the cyanobacteria, actually, which is kind of, what we would expect. And then we have, some other phyla that, if you really care about freshwater bacteria, you'll get excited about, but probably not going to have time to go into very many of them today. But just to give you a sense of the statistics here, so I actually don't have a final count on the number of reads that we had, but we have on the order of 10 terabase pairs, and then we did individual sample assemblies. So assemble each of the 460 samples, and so then you get about 430 million contigs out of that. Then you do an all versus all read mapping, and this is the really computationally intensive part. So we use a mapping algorithm, we use Bowtie for those of you who do this stuff, and compare all of the metagenome read sets from all 460 samples against all of the contigs, right? So it's a big all versus all mapping. And then you sort it, you put it through some pipelines that people have developed. And I got about 14,000 bins. And then you have to do some quality control on those and sort out the bad ones and then de-replicate. And so this is a really important step. We decide, because I see the same genome over and over again if I have 460 samples, right? So I have to get rid of that redundancy. And so I ended up with 1,100 and something non-redundant genomes. And the definition of non-redundant is 98% genome-wide average nucleotide identity, right? So you have to make that decision. And we'll talk about that. I did have some single cell genomes. So the students who read the papers that I passed along, those were about the single cell genomes that we had in 2017, 2016. And so we added those in too before we dereplicated. And so we ended up with just, a little bit more than 1,000. And then I said, I can't handle this, this is too many. So I'm going to only look at the ones that are found in at least 10 samples with 5x coverage, right? So I end up with this like golden set of 826 reference genomes. This is just the top 50, okay? So you can see there's a lot of rare ones. So a lot that are rare in the data set, either because they only appear a few times, a few days, or because they don't have they're low abundance, they have low coverage, right? So you can take these genomes and do the same thing, basically, that Robin did and looked at community ecology questions, because now you just have another way of measuring the abundance of that species, except that you're defining the species as this non-redundant genome that you can map reads onto and get an abundance for. And so, we have to do that. We're going to do that. I haven't done that. I think it's kind of, I shouldn't say it's boring. It's not the most exciting part that I want to do with this data set. And so I want to find other ways of using the deep metagenomes to think about processes that are happening at timescales that overlap ecology and evolution, right? So kind of an eco-evo approach. And the way that we've been thinking about this comes from very early metagenomics. This is a kind of plot that you used to see all the time in papers at the very start of short read metagenomics. So what we have here, it's a little hard when the first time you see it, but the x-axis really is the genome, right? This is an incomplete genome, so it's like just the contigs in a random order. There's like no significance to the order in which the contigs are lined up, and it doesn't even show you where the contig breaks are. And then the y-axis is the percent identity of the short read from the sample that mapped against it, right? We call this a recruitment plot. So back when we used BLAST for doing this sort of thing, we would use BLAST to recruit a read onto the reference genome. Nowadays, we call it mapping, right? We just had different words for it back then. And reads that map at 100% are presumably like identical to the genome. They're from the exact same strain, population, like whatever you want to call it. And then you've got this smear of reads that are at a lower percent identity. And so you ask, well, which of those organisms that contributed those reads are part of the same species or part of the same, whatever taxonomic unit I want to use. And people noticed that when you made these, if you squint, you can kind of see that there's fewer reads in this region of the plot. And it's a little hard because once the reads start, or the dots start plotting over each other, then you can't see how many there are. But I'll turn this plot on its side in just a moment. But what we think, just to kind of give you the punchline, is that there are coherent populations above around 95% nucleotide identity. And below that, you have related organisms, but there's something that is keeping them separate. And, if you believe in biological species concepts for microbes, then you can say, well, maybe there's some barrier to recombination so that we can't get, any kind of exchange between, the populations that are represented by these reads versus these, right? But that's all, like, people haven't really studied that in natural populations really at all. And so I promised that I would turn it on its side. So this is what the same recruitment plot looks like, but I now have the percent identity on the x-axis, and just it's a frequency plot, so the density, number of reads that are at each of those percent identities. And so this is one genome with one metagenome mapped against it. And so you can see that there's reads in that metagenome that look like they're nearly identical to that reference genome. And then you have that discontinuity, right? And then you have a whole bunch that are maybe from a different genus or don't even know, right? And so we looked at this a lot. Some of these plots are in the Garcia 2018 that I shared with the students. And what we found was that, and in hindsight, like this should have been kind of exactly what we predicted, but we didn't really know that if you do this with a genome from this, in this case, a genome from Maine, like the state Maine, and map a Mendota metagenome on it, you don't see that hump of very highly related reads, right? This implies that there's some kind of geographic separation such that there's, not an identical genome in Lake Mendota. Or maybe the lake in Maine is just really different from Lake Mendota. We actually don't know. We happen to get this single-cell genome from Ramona Stepanauskas, who was a big single-cell guy. Back in the day when we were doing this. Here's one that comes from Germany. So that, again, the genome is from Germany and the metagenome is from Lake Mendota, which strangely enough is farther away than Maine, but still has some like slightly more related genomes in it or population in it. But this kind of taught us, like, we don't want to do that anymore. We don't want to take a metagenome from one ecosystem and compare it to a genome from a different ecosystem, because we don't really know how to interpret that discontinuity in that situation, because we don't understand migration, we don't understand geographic like isolation in terms of gene flow, and so we're just not even going to do it. So now we just use genomes from Lake Mendota and compare metagenomes to Lake Mendota. So there's a limit to how you can use the genomes for this purpose. I did want to show this one because it's so strange. This is a particular group of freshwater bacteria that are related to Pelagibacter ubique. So if you're an aficionado of the ocean, the most abundant bacterium in the ocean is called SAR11, Pelagibacter ubique, and it has a sister clade in freshwater called LD12. It's since been named Fonzibacter, and it has a very strange population structure. So you can see that there's not very much of a discontinuity, and it kind of has reads that map at sort of in that forbidden zone, and not very many, far out at lower percent identities. And so it's interesting to think about, like, what would be different about this particular group versus some of the ones that I showed you in the previous plots, because this is really the exception. The other ones all kind of look more or less like the other one. So this one is the weird one. And so you can think about transitions from saltwater to freshwater and what might be constraining gene flow there. And those are all really interesting questions that we haven't been able to study yet. So assuming that we're all comfortable with the idea that this discontinuity means something, right? And that it's a good place to demarcate what we're going to call our new unit that we're going to count with. We can say, I'm only going to take the metagenomic reads that map to a reference genome above, 97% or 98%, whatever cutoff you decide you want to do. And that's where that dereplication cutoff comes from too, that 98% that I decide I'm going to say this genome is distinct from that one, but I'm not going to look at all the genomes inside of those clusters. And so trying to be, consistent in how we define that. And so I told you already about all the genomes that I found. I want to give you, some names so that you have some sense that you know them, and how they relate to, processes that you might think about yourself. This is a slightly more zoomed in rank abundance curve. This is the 16S, but it gives you the same names. So the top two organisms here are actinobacteria. So you think about actinobacteria, you think what? Anybody think about actinobacteria in the room? Huh?
Soil.
Oh, that's a good one. Sure. I think soil, right? So soil and antibiotics, right? Like that's where we get a lot, streptomyces, right? We get a lot of the antibiotics from them, right? These organisms are, they look like a streptomyces where you took away all its secondary metabolite machinery. They are really small genomes. They're about 1 megabase pair. So like pushing the limit of what you can get for free living organisms. And they are free living. You can see them under the microscope using fluorescent in situ hybridization. We know they live by themselves. They are also numerically the most abundant taxa in the lake. And then you start seeing cyanobacteria. You see some, these degrade polymers. These are those weird LD12 organisms. So you can kind of go down through there and pick out your favorite organisms and start looking at their genomes. And I color-coded this by the ones that are found in all the samples and at some, arbitrary threshold above which we think that it's important. So I'll do a little vignette here on those top 2 organisms, just to give you a taste of where we're going. And we call them AC1 actinobacteria. And more recently, there's been some analysis. I forgot to put the name of the paper here, but this is not us. This is a group on the Czech Republic, but trying to culture them and give them names. They have very mixed results culturing them, but there's basically two major clades. There's the A clade and the B clade, and then within those, there's diversity. There's more diversity in the A clade, so maybe you can see just in terms of the branching order and the lengths of the branches that these are the more diverse within the clade. And they are everywhere. They're cosmopolitan. You find them in every lake you've ever looked in. And they also are very active in terms of their transcriptome, right? So they're not just abundant, they're also active. And this is a figure from the paper that I sent to the students just to kind of link this back to that original work that got us thinking about this, that you've got your AC1, B1 up here, and I'm showing the time series that we had at the time that we wrote this paper, and the color of the dot shows the percent, the average percent identity of the reads that mapped to it, and the abundance is the diameter of the circle. So there's this very abundant clade or very abundant strain that we didn't probably have the exact genome for because it's pretty low percent identity on that scale. And then you had these 2, which are members of the same sequence discrete population that had an interesting pattern where their diversity dropped towards the end of the time series. So we can look at these more closely. Now we have more genomes, we have more metagenomes, we have deeper metagenomes. So these data sets were much smaller than what we had before. I don't want to spend too much. I'm going to skip through a few things here. I can save them for when I talk to the students though. So these are the genomes that I have that are just from the AC1 group, which has now been renamed as the family Nanopelagic ACA. But so not a huge number, but this is, the non-redundant set. And you can see the rank abundance curve. Again, that's what I want to draw your attention to. So the x-axis is just an overall abundance in the data set, if you will. And if you can squint, you can see that the most abundant one is an A, and then you get another one that's a B, and then you get another A, and then a collection of Bs, right? So, and these are distinct enough that they were non-redundant genomes. So we did capture a lot more diversity than we had in the Garcia paper, right? And that was exciting, of course. And I was curious about linking these new genomes back to what we saw in the Garcia paper by looking at changes in the percent abundance over time. And this also kind of relates back to the 16S work, but using a finer scale of resolution. And I have very novice R skills. So they made this in R like 2 hours ago. So I'm apologize if it's not very beautiful, but like I'm having a lot of fun getting ready for this. So these are all my genomes, and this is the year, right? So I have now aggregated their abundance by year. And so you can see that there's some genomes that, were there all the time, and others that have kind of come and gone, most of which are A's, actually, AC1A. I can't explain why they've gotten so much more abundant in the later years. What I suspect is that there are other organisms that have gone down in abundance, because these are all relativized measures, because you can take a metagenome and map it against a collection of genomes, and you divide by the total number that mapped to get a percentage. Because if you don't do that, the size of your metagenomic sequencing effort, affects that number. So you have to normalize to the total amount that you had to start with. And so something else is going down in the lake. And I don't think it's the actinobacteria. I think it's something else. But I saw this like 2 hours ago. I was like, oh, that's cool. So we'll have to follow up on that. And we also have a new way of identifying or measuring the average nucleotide identity. So this is using a software package that gives you nucleotide diversity. So for those of you who do population genetics, that's pi. And so this is the average pi for each year for each genome. And what I was expecting to see was that one of the genomes that would correspond to that sequence discrete group that I'm talking to the students now because the rest of you probably don't remember, but in that previous slide, though, where the diversity increased, increased in more recent years. I kind of expected to see that, and it might have something to do with the scales of the color, but I don't see it here. And so it definitely makes me want to dig in more to slicing and dicing those interannual patterns to see if there's a shift, or if it was just because of the reference genomes that we were using in that data set, or because there's like a million data analysis regions why it may not have been real, or why this might not be real. So we have to dig a little harder into that. Yeah, sure.
Is increasing in population size abundance?
I have a well, so right. Defining population size here, I think, is really hard if you just use abundance as a proxy. Well, this is the plot of abundance versus pi. So there is a relationship, but I suppose if you cut, it's almost like if there's a threshold where it becomes like yeah, and not very much. People haven't looked at this in natural populations because they've looked at cultures, but there you're looking at many fewer individuals. And so some of the ways that we think about it, I think we have to figure out how to wrap our brains around it. Yeah. And here's one thing that'll make your brain explode. So I've stepped away from thinking about just the actinobacteria. I've gone back to my list of the top 50 genomes. And here I'm plotting, this is my last data slide. So if you're feeling saturated, it's okay. This is the distribution across the entire data set for each genome of pi, right? So like for every sample underlies this bar plot, it's not just in years. So if you have a high average, then you're over here, right? And if you have not very much variation in your pi, then your box is, narrower. And I just wanted to look at what the distributions behave like
across years, or
this is across the entire data set, so like all 19 years. So like each of 460 samples is an individual observation that goes into the box plots. So you can see that at the top of the list is that LD12 and the AC1B1, which means that they have, across all of them, those genomes have the highest level of nucleotide diversity. So they are the most diverse within their populations. And then, you can start looking down here, you see a lot of the cyanobacteria, which kind of makes sense because they are boom and bust, right? They bloom and then they crash. And so you would expect that there'd be low diversity in their populations when they bloom. There's a few exceptions up there. And then this one always jumps out to me, and we don't know anything about those organisms. Pirelliaceae, they're like planktomycetes, which nobody studies. And they have extremely variable nucleotide diversity. So they are like, their populations are kind of doing this over time in terms of the amount of diversity they have. And so the next thing I want to do is to dig in some of the extremes here and figure out like what's so different about the LD12 and the AC1 and B1 versus maybe some of these, maybe the non-cyanobacteria, because there's a lot of down here, and then what's up with those weird planktomycetes. So we're just sort of at the beginning of trying to understand, what this means. But I think it creates this really neat opportunity to look at population genetics and population structure in natural communities with the time series factor in there at different time scales and see how the genomes are changing and diversifying and potentially losing diversity through time. And this is the new stuff that I'm like, oh, somebody tell me if I'm on the right track. I think with that, I'll jump to my acknowledgements. So Robin really was the driver of the initial data analysis. She's also asking really similar questions to what I'm doing myself, but she's doing a postdoc now, and so she's working on that. We're kind of working on that together. And then my longtime collaborator at Stockholm University, who thinks a lot about that same actinobacterial group, Sarahi Garcia, who is the lead author on the paper that the students read, and then a bunch of collaborators, especially the JGI, because they've been incredibly supportive of us over the years. And yeah, I went a little over, but hopefully we have time for questions. Thank you. Yeah.
Nice talk. I'm really, I thought it's really cool how you combine population genetics with metagenomics. It's not an uncommon thing. So that's really cool. So I was just wondering, these types, to what extent do they vary in their mutation rate? Because that would affect I.
We have no idea. No idea. I mean, they aren't in culture. None of these are. I mean, some of the cyanobacteria are in culture, but like that long list are all just weird uncultured things that are in lakes.
Oh.
So we don't know. I mean, that would be one of the things I'd want to try to figure out.
Yeah.
One of the problems with short read metagenomics doing this is that you don't get linkage.
Yeah.
Or you can try, but you don't get very much information about it. Right. Yeah. So that is a big limitation.
Okay.
Yeah.
Are there caveats to using reference genomes even within the same like if you're looking at for time or like seasons or from year to year, is there enough variability that there are variables down the road?
Yeah. I've thought a lot about that because you dereplicate partly for your sanity because you can't deal with so many, because it reduced the number by about 1,000 or 10-fold. But you also do it just because you're not really sure when you're dealing with two members of the same population genome thing that, like if you map reads, are they going to go to both? Or are they going to pick one when you map? But I think about that a lot. It's like if I chose a different reference genome as my representative, would I get a different answer when I'm working with a reference? And I haven't gotten to looking at that yet, but that does make me worry. But I also haven't thought of a way around it, right? You have to pick a single reference because of the mapping. Yeah.
I was just wondering, for the species that are not always around, the ones that come and go, are you able to see if there are either positive or negative correlations between different groups? Because maybe they're collaborating and they're inhibiting each other. And along those lines, do you also have data for all the range of years for how the chemical composition of the range is changing, so you can add that to the correlation and try to figure out biologically what all this might mean? Yeah,
for 16S based work, there's a lot of tools out there to do that, like ways of looking at correlation networks to see taxa that are affiliated with one another through time. And some of that work has given us clues about specific groups, like the Bacteroidetes I mentioned earlier is a group that breaks down polymers, and they track the cyanobacteria. So, cyanobacteria bloom, they die, they start falling apart, and then the ones that like to chew on them, come up in the data set next. So there's some of that's pretty clear. You can do a lot of that. In the end, it's kind of like, here's a bunch of correlations and you're not really sure. It's a hypothesis generating exercise, right? Because you want to then think about experiments that you could do to make sure that wasn't just, are they interactions or are they both responding to the same environmental driver, for example. The chemistry data that we have is, I don't know, when we write grants, we're like, we have all this great chemistry data and we can relate it to the population dynamics or the community dynamics. And then when we actually sit down to do it, it's a little bit unsatisfying. And the way I try to describe it is like, if you measure a certain amount of phosphorus in the water, right? Okay, so phosphorus is a big driver of cyanobacterial populations at the ecosystem scale. So you measure phosphorus in the water and you see it's going down over the summer. But if there's no phosphorus in the water, that doesn't mean that they're not getting phosphorus, because there's a really high turnover rate. So as soon as somebody breaks down and releases their phosphorus, it's sucked up. So you never see it, right? You're not measuring the flux of an element through the system. And so the problem with those measurements is that it's not really measuring what's available for the microbes to use. It's almost like when you go to beer fridge and you open the door and there's beer in there, that's probably the beer that nobody wanted, right? Because all the rest has been drunk. So we struggle with that a lot though. But yes, we have lots of chemical data and it's great. And you can correlate it to all the microbes, but has its limitations. Yeah.
So if I remember correctly, I thought you mentioned the zebra mussel increase is associated with the diversity of the cyanobacteria. Did you say that?
Yeah, it seems like it's also the spiny water flea and the zebra mussel, or maybe it's just the spiny water flea, because we can't really.
Are they correlate? I mean, affect each other, or just responding to the environmental change or temperature change independently?
Yeah, there is a whole set of ideas about that Jake van der Zanden has related to, like, what do they call it? Sleeper populations. So like where the spiny water fleas, they got a foothold because we had a very cold summer, relatively speaking, and they like cool temperatures. So once they got going, they had a population size big enough that they could maintain themselves. And so Jake's group thinks that that somehow facilitated the eruption of the zebra mussels, because the zebra mussels were there before. They were just present at very low numbers. So that there was something about the change in the nutrient cycling or the food web structure or something that the spiny water flea caused that allowed the zebra mussels to go nuts. How does they have veligers? Like, do you think that the spiny water fleas were eating the veligers? Yeah.
They could, they're filter feeders.
Mm-hmm.
So that would help. I mean, the thing is, zebra mussels are originally from a more saltier environment, so they don't like things to be super.
Oh.
So they like higher ionic concentrations. They could be getting nutrients.
Yeah.
They're not efficient freshwater animals, so they could be getting nutrients from the veligers.
Well, and I mean, we definitely are putting a lot of salt on the roads. There's that. I never thought of that. Yeah.
And they adapted, they evolved, but they're still really inefficient as freshwater osmolators. They're very leaky.
Crazy. I didn't know that.
Yeah.
Huh. I have to think about that more.
Hmm.
Cool. But, as always, we also really wish we had more data, like more, the invisible present is a problem because we only have 3 years of the zebra mussel post-invasion. And the lakes have actually been relatively clear in the past 3-4 years, and like not very many cyanobacterial blooms. So Maybe, but they don't have a lot of hard surface, so their population densities are pretty low, except on the edges.
Are there any bugs? Bugs can be bug.
I don't think that Jake has found any of those. But he also hasn't been looking because he doesn't have funding right now specifically to do that. Thank you for listening.